Good afternoon. My name is Sori and I'm a master's student at the Evolutionary Studies Institute in South Africa. And today I'd like to present my research titled The Role of Death in the Rise of Eukaryotes, the Case for Archaeal Caspase Homologs. So eukaryogenesis is the evolutionary process leading to the origin of eukaryotic cells, and it's one of the major transitions in the evolution of life. So it's basically the emergence of the first eukaryotic common ancestor Fika, which led to the development of complex and different eukaryotic cells. And the most commonly accepted hypothesis of how Fika arose is via an endosymbiotic event between an archaeon and an alpha proteobacterium between 3.48 and 1.7 billion years ago. And the period I would like to focus on is this period known as eukaryogenesis. So if I explain this further, this is a simplified hypothetical picture illustrating the concept of endosymbiosis, and a major problem in this scenario has been the difficulties that arose post-engulfment of one prokaryotic cell by another due to competition that arose between these different individuals. So the problem was due to the different evolutionary interests of these individuals that weren't aligned, but stable eukaryogenesis could have only occurred if these initially independent units interacted cooperatively. So it's expected that in this kind of major transition, the symbiotic relationship between the archaeon and the bacterium would have constantly gone through cycles of cooperation, conflict, and conflict mediation, and for them to remain in a stable union, this conflict would have had to be mediated and resolved. And numerous hypotheses have been proposed as to how conflict was mediated, and programmed cell death, PCD, is proposed as one of the mechanisms, where death would have enforced cooperation between the bacterium and the archaeon by genetically aligning the different organisms. So to explain PCD is a genetically programmed cell death, and a whole range of genetic toolkits are required for the molecular pathway. And one of the main effectors of PCD are the caspases that are activated by both the external stimuli and the mitochondrion, but caspases are known to be only present in the metazoa. But now through research, we know that there are a lot of caspase homologs that are present in prokaryotic organisms, such as orthocaspases and type I metacaspases present in bacteria. And this classification is obviously based on their domain organization. So studies on PCD and origin and how they participated in endosymbiosis have been historically limited to bacterial lineages due to lack of empirical evidence of PCD in archaea. And this is especially due to the lack of genomic sequences available for archaeal organisms compared to bacteria. So with the additional archaeal genomic information available, with the advances in metagenomics and identification of archaeal lineages, we wanted to know if this genetic toolkit is also present in archaea, and if PCD did indeed act as a conflict mediator during eukaryogenesis, we wanted to investigate the origin of this main effector of PCD with the newly available sequences. Therefore, my research question was, are caspase homologs present in archaea, and what does this mean for caspase homologs acting as a conflict mediator during eukaryogenesis? So, for my methods, I first obtained protein sequence data from GenBank, and then I confirmed them as caspase homologs using HAMR and inspection of the HC catalytic dyad, which is a prerequisite for caspase homologs. Then I looked at the taxonomic distribution and phylogenetic reconstruction to refer to their history. And this part was particularly important because eukaryotic caspase homologs are suggested to originate from bacteria during endosymbiosis, and I wanted to see if this was the case for archaea as well. Lastly, I did a phyletic pattern analysis, which refers to protein domain and domain architecture analysis to infer the ancestral function. And the filtering step of ham search and the HT diet inspection revealed 230 sequences to be confidently predicted as caspase homologs, specifically the metacaspases and the orthocaspases. The distribution of sequences according to different phyla showed that most sequences were identified in the phylum Eukaryotes, but sampling bias is suggested in the results, as archaeal organisms of other phyla are very challenging to culture and sequence. And because identification of caspase homologs in archaea is a novel finding, the distribution of different members of the family, so the caspases, metacaspases, orthocaspases, and paracaspases, were mapped onto the tree of life. And as you can see, paracaspases and caspases were only present in higher complex organisms, but orthocaspases and metacaspases were mostly found in simple unicellular organisms, and their presence in deep branching lineages of archaean bacteria suggests that caspase homologs are ancestral proteins, and this suggests that it is more likely that these caspase homologs were present since Lucas, with large gene flows between them. So next we wanted to look at the validity of this statement through phylogenetic reconstruction. So this is the evolutionary history of orthocaspases in all three domains of life. And the question we wanted to ask was on the origin of these archaeal caspase homologs and whether they were present in ancestral organisms prior to the rise of eukaryotes, or whether they were acquired via horizontal gene transfer from bacteria. So as you can see, the phylogeny of orthocaspases revealed a scattered distribution and did not follow the classic 16S rRNA tree. And this was identical for metacaspases. So for both trees, the deep branching caspase homologs belonged to bacteria, but archaeal caspase homologs were still placed near the root of the trees. So identification of orthocaspases and type of metacaspases in deep branching phyla of bacteria and archaea, and the incongruency between the species tree and the gene trees suggests no single evolutionary event is responsible for the observed distribution, and that it'll be difficult to deduce their common ancestor. So based on this observation, we hypothesized that ancestral orthocaspases and type 1 metacaspases were present since Lucas, and this would explain the scattered distribution and the incongruency, as these caspase homologs would have been present since Lucas, followed by the emergence of bacteria and archaea with large-scale gene flows between them. So the main conclusion is that the principal PCD molecular machinery did exist in archaea as well as in ancestral organisms since Lucas. So the conservation of these caspase homologs implies their importance, and their primitive functions during eukaryogenesis is open to discussion. So we wanted to investigate the potential function of archaeal orthocaspases and metacaspases through phyletic pattern analysis, basically their domain and domain architecture. So the presence of the C14 peptidase domain within a caspase homolog does not guarantee the participation of PCD. Additional protein domains are required, which would act as receptors and adapters of the cell signaling pathway, also known as the domains of death, as seen on the diagram. So a lot of different protein domains were identified that are part of the domains of death, but a lot of other protein domains associated with cell survival were identified as well. According to the domain architectures, multiple caspase homologs resembling a signaling peptide responsible for interacting with the external environment was observed, where they possessed a transmembrane domain, followed by an extracellular C14 peptidase domain and a domain associated with cell survival to adapt to surrounding environments. Interesting cytosolic caspase homologs are also identified that possess the death-associated domains, but these domains are also associated with non-PCD roles, and further in vivo analysis is required to deduce their molecular function. So the evolutionary history of protein domains present in archaeal caspase homologs proposes a support for the original sin hypothesis of origin of PCD. So origin of PCD has been debated a lot, but a philosopher called J.C.M. Mason suggested there is no bona fide genetic death program in our cells, and it actually originated from a combination of different gene products participating in different pathways involved in cell cycle and cell differentiation, present since the origin of life. So as in line with the hypothesis, you can see that also caspases associated with cell survival were present in ancestral archaea, and with the rise of late branching superphyla of archaea, caspase homologs possessing death-associated domains started to appear. And at the point of eukaryogenesis, it is likely that archaeal caspase homologs possess death-associated domains, which were later integrated into the PCD molecular pathway. In conclusion, this study has shown that caspase homologs are present in archaea and that they are present in all three domains of life. Based on this data, I speculate that the main effector of PCD emerged very early on, and the phyletic pattern analysis suggests they were survival-related and later death-associated domains were required. So elements of PCD are present in both prokaryotes, and potentially both the archaean and the bacterium were genetically primed to induce death in itself and in others. So in conclusion, this research has provided the first layer of information to determine if PCD did indeed act as a conflict mediator during eukaryogenesis. Thank you.